Hello everyone. In the different environments that chemical reactions can occur, there can be many other substances that can impact enzyme-catalyzed chemical reactions. For example, our own bodies, which are host to many chemical reactions that are catalyzed by enzymes, always have many compounds within the tissue and bloodstream. We can characterize some of these substances and how they interact with enzyme-catalyzed reactions as inhibitors. Inhibitors are generally proteins. There are many types of inhibitions that exist. For our purposes in this course, we will be focusing on competitive, non-competitive, and uncompetitive inhibitors. The difference in the types of inhibitors is how and where they bind to the enzyme to impact the enzyme-catalyzed reactions. First, we will be talking about competitive inhibition. Competitive inhibition occurs when the inhibitor will directly block the active site on the enzyme to prevent the chemical reaction from being catalyzed. This means that there is a presence of the enzyme, substrate, and inhibitor. There is two outcomes resulting from the enzyme and its interaction with another substance. It will either form an enzyme-substrate complex, as shown as this image as the ES complex, or an enzyme inhibitor complex, as shown as this image as the EI complex. However, it is important to note that the enzyme-substrate complex will be the only complex that will create the product. Second, we will be discussing non-competitive inhibition. Non-competitive inhibition can also be called mixed inhibition. Non-competitive inhibition occurs when an inhibitor doesn't directly bind to the active site to prevent the substrate from binding, but rather binds to another part of the enzyme that can then prevent the enzyme from catalyzing the substrate to make a product. During non-competitive inhibition, there can be three types of complexes that can be formed. The enzyme inhibitor complex, also known as the EI complex, the enzyme substrate complex, also known as the ES complex, and the enzyme substrate inhibitor complex, also known as the ESI complex. As you can see in the image, we've shown where each stage has the EI, the ES, or the ESI complex. If the enzyme and the inhibitor first bind in the reaction, then the EI complex is formed. The S can further bind to this complex, creating an ESI complex. This ultimately leads to no product formation unless the inhibitor is released and the ES complex is formed. Another route is that the E and S can first bind to create an ES complex, but if an inhibitor binds to the ES complex, then again the ESI complex is formed and the product cannot be formed. However, if it doesn't bond, the product can be formed. So it's always important to remember that the only complex that can produce any product is going to be the ES complex. Finally, we will be discussing uncompetitive inhibition. Uncompetitive inhibition is when one or more of the substrates must bind to the enzyme before the inhibitor can bind. This means that the two complexes can only be formed: the enzyme substrate complex, again the ES complex, or the ESI complex, or the enzyme substrate inhibitor complex. But because the substrates must bind to the enzyme before the inhibitor can bind, the ES complex must be formed before the ESI complex forms. If the ES complex does not have the I bind to it, then it can then proceed to create product. However, if an inhibitor binds to the ES complex, this means that the product cannot be formed unless the inhibitor is released from the ESI complex and the ESI complex turns into the ES complex. Having an understanding of the different types of inhibitors is important for modeling purposes because if we are working with inhibitors, we must know what type of inhibition it has with the enzyme, as this ultimately affects how we model the product formation, or how much product can be made from the presence of the inhibitor. One thing that you may be wondering, based on this lecture, is how do we find out what type of inhibition an inhibitor performs? Usually, you can research papers that determine the type of inhibition for the inhibitor in interest. The methods that papers are able to find the type of inhibition through is using an enzyme assay and then further analysis of the inhibitor activity within the enzyme-catalyzed reaction. In an enzyme assay, scientists will first be able to find the important values for the Vmax, or the maximum velocity that a product can be formed from the enzyme and substrate being catalyzed without the inhibitor, and the Michaelis constant of the substrate, which is the concentration of a given substrate which catalyzes the reaction at half the maximum velocity. Then, the scientists can observe the changes of the Vmax and the Michaelis constant when adding the inhibitor to the enzyme-catalyzed reaction to ultimately determine what type of inhibition the inhibitor displays. If you would like to read more about this process, we will put a link to a paper that clearly explains what patterns will indicate what type of inhibition will occur based on the changes of Vmax and the Michaelis constant. Thank you for watching this video.